Check, 1, 2. To use this, you just push and the light turns on when it's on. There we go. Okay. If it does that blinking thing again, just give it a good look.
Okay.
Let's go.
Check, 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 1, 2. Look at me go.
Green should mean it's working. I think I can hear it.
Can you hear me up there? Samantha, yes? Okay, okay.
Yes, this is working for audience questions. Right. Maybe. Hypothetically, just needs to take a nap as well. Me too. I'll break it by syncing with everyone.
Anyone with IT knowledge is worth their weight in gold, right?
All right, can you all hear me? All right, so very quickly, I'm here to introduce Dr. Heather Dunn, who is visiting from Clemson's Bioengineering department. Her work is inspired by previous postdoc positions she had at Dana-Farber, where she was responsible for coordinating a clinical trial that looked at combination therapies for those triple-negative breast cancer patients that had lung metastases. And she's speaking today on translating breast cancer mechanisms from ethnically diverse samples. Before we get started, I do have an award to give you very quickly for coming here and speaking with us, the Cancer Warrior Award. So both welcome and congratulations to Dr. Dunn.
Wow, thank you. Thank you so much. Well, that is wonderful. Thank you. What an honor. Thank you so much. That's beautiful. Good. Volume's good. Thank you for the opportunity to be here today. I appreciate it. I'm glad to be here. And I hope you'd gain some insight from my talk. My goal is always for everyone in the room to at least learn something. So maybe you might not learn something about science, but hopefully so. So yes, the title of my talk is obviously translating the breast cancer mechanisms from ethnically diverse samples. So this is probably a review for most of you, but my work is focused on triple-negative breast cancer. And triple-negative breast cancer is diagnosed in almost twice as many African-American patients as it is those of European-American descent. In addition, they have approximately a 40% increased mortality rate. They also are diagnosed at a younger age. So it used to be the general consensus was, well, the reason for this disparity, it is because of social determinants of health. And while we may have some environmental factors that do play a role in this, certainly there's got to be something else other than the social determinants of health that lead to this almost twice the increased incidence compared to other racial groups. So work in my lab has really been focused on going forward in trying to prove, or rather disprove, that it's not just all social determinants of health, that there are also some biological factors involved. And one of the works that we did, and we published this back in around 2022, 2023, we did a project where we actually, and we started this during COVID, and we're like, okay, what can we do since we can't be in the lab? And so we visited the TCGA website and we downloaded images of tumors from patients that had self-identified as either Caucasian or as African-American. So we have these images, we have basically just H&E images of these tumors. And we downloaded approximately, and I think the numbers were around 117 from African-American images, unique patients, and approximately 130, 135 images of these tumors from the Caucasian patients. Now, and keep in mind, these are not just triple negative. Okay, so we have different subtypes here, but the main thing what we were going for is we were looking for tumors that were from patients that had identified as African-American. If we filtered out for triple negative, we had a very small sample set. And so what we did was we took these H&E images and we programmed our computer. We decided we're going to test this AI model and see if this is going to work. So we took 80% of the images from each racial group and we provided to the computer. And we trained our model and we're like, okay, these images are from African Americans, these are from Caucasians. And then with the remaining 20% of the images from each racial group that the computer has not seen, we provided the images to the computer and we're like, okay, computer, now you tell us which racial group these belong to. And in AI, you know, if you get over 70%, that's pretty good, right? That's some pretty good data. 80%, I mean, it's to be so. And anything over 90% is just mind-blowing. So I have the first model that we ran, and the order of these are the different orders that we ran the models. So the first model that we ran, it was AlexNet, and we ended up with 92% accuracy. So in other words, the computer was able to detect which racial group these tumors came from just by viewing an H&E image. And we honestly thought it was a fluke, and we're like, this can't be right. So we did it again, and so we did it with the second model. So again, we did ResNet 18, and as you can see, we went through these 5 different programs to analyze the data. And we had 2 of them that had 92% accuracy, and our lowest accuracy rate was 84%. Now, people say, well, clinically, how does this matter? Well, it doesn't matter clinically. I mean, obviously, we can just ask a patient, you know, which racial group do you identify with? But what it tells us is that morphologically, something's different, okay? So then our next challenge was to figure out, okay, what is happening? So what is the computer? What does it see that we can't see that the differences are? And so the first explainer method that we ran, and it's called LIME. And what LIME does is it ends up putting this green spot over tissue areas where it's detecting a difference. So basically what we had to do was analyze what was underneath that tissue. And we found out with this explainer model that the majority of the tissue differences across these 2 racial groups was the interface of the tumor and the extracellular matrix, okay? Or basically the microenvironment that is surrounding this tumor is where this computer model is detecting a difference. So we ran and which was wonderful because we thought, okay, this is kind of where we're wanting to go. But we ran a second model. I'll go back. And this is a saliency map. It's kind of difficult to see exactly what this is looking at, but the saliency map explainer is evaluating pixels. So it's on a pixel basis where these differences are when we were comparing these 2 different groups. And again, it identified the tumor, where the tumor and the extracellular matrix or the tumor microenvironment at that interface. So let me just kind of go through just this. Hopefully you all are aware of this, but we have the tumor, right? And then we have this entire region around the tumor. And in speaking with oncologists, you know, they say, you know, we have been sampling the tumor and we biopsy the tumor and we decide on our treatment based on what the tumor is. And then after, you know, I talked to them about this different data, they're like, maybe we also need to analyze the extracellular matrix and this tumor microenvironment. Maybe we need to see what this niche is outside of this tumor in addition to just testing the tumor, because maybe we're chasing the wrong thing. So where are all these cues coming from that are driving these tumors? And some are definitely coming from in the tumor itself, but then there's also some cues that are happening out here in this microenvironment. And then another question is does the microenvironment activate cells that are considered dormant? And I just attended a couple of months ago a Keystone conference, and the whole conference was based on tumor microenvironment and its role in metastasis. So it's quite interesting where I think we've shifted some of our focus away from not only just the tumor cells, but also what is surrounding that area. And another thing, this is just another example where when we talk about this whole tumor microenvironment, we have the extracellular matrix, right, which is a lot of the support network that we have. Then we also have the stroma and these cells that are involved in it. And then we also have these immune cells that are playing a role. And I want to just tell you just a quick story about immune cells and the incidence of racial disparities in disease. There's a scientist, and she's at the Medical University in South Carolina. Her name is Marvella Ford, and she conducted a study where she was analyzing breast cancer patients, and she used this subpopulation of people in it's called the Sea Island natives. So it's basically a small barrier island off the coast of South Carolina. And the people that live, these natives that live on this island, are more closely genetically related to African descent than they are to African American. And so she evaluated the breast cancer incidence and the tumors from these people that were these Sea Islanders. And then she also evaluated tumors from Caucasian individuals and African American individuals. And what was interesting is the Sea Island native islanders were more resistant to aggressive subtypes than African American patients are. And she has come up with this theory that African Americans are, and this is generally speaking, are more immune suppressed than people who were African descent or European descent, because she's saying that they are a younger generation or a younger population, that they've only existed for a few hundred years. So maybe their immune system is not as robust as other ancestral groups, because the African American, and she gave me a phenomenal history lesson, which I think I needed. She was saying that when slaves were brought over from Africa, it's very expensive to go purchase more slaves and bring them over. And so as an African slave would have a child, right, the slave owners had another slave. And also, which I did not know this, she said that the slave owners, that a lot of the sons of the slave owners could have their way with any of the slaves that they had. And so a lot of young men came of age, if you will, with the slave women, and they decided that any child born of a slave would assume the ancestral group of the mother. So she was saying that this is actually how a lot of our African-American race originated. And so, I mean, we're all diluted factors of where we're from, but I thought that was very interesting with this whole immune subponent of the entire extracellular matrix. It is, I think it's something to further investigate. So one of the studies that we started and to evaluate this tumor and this microenvironment interaction, what we did was we obtained patient samples. These are tumor samples that were collected from diverse patients. And in this study, we had 5 that were from European American or Caucasian and 5 from African American. And we took these samples. We had a pathologist score all of our slides to denote where there was actually the tumor and then where we were outside of the tumor region. And utilizing laser microdissection or LMD techniques, what we did was we lasered out 4 distinct regions. So the first region is, so if we're going from the left to the right, the first region is the center of the tumor, right? Then the next region is the tumor. It's the edge of the tumor, but it's still the tumor, okay? It's all tumor just right there on the edge. The third region is the extracellular matrix, or the tumor microenvironment, as close to the tumor as possible. And then we try to establish our own internal control, where we basically collected from the extracellular matrix as far away from the tumor tissue as possible, right? And then what we did was we conducted RT-qPCR on all of our samples. We analyzed 40 different genes. We had a few housekeepers, and most of our genes that were selected were highly implicated in the EMT process, right? And what we found out, and I have to tell you, it took us, we just had this accepted for publication like January 30th of this year. And it took us several years to get this work published, because a lot of the reviewers kept saying, well, you only had an N of 5 from each racial group. But we finally pushed, and we were determined to get this work published. But what we found, which was quite interesting, is if you look at the PCA plot, so we have the cluster, so this red and deeper maroon, that is the center of the tumor. And then that is the center of the extracellular matrix. So we have a lot of overlap in the cell population there. And then the other 2 circles that we have, the green and that blue, the green is actually the edge of the tumor. And then that blue circle is the edge of the extracellular matrix, okay? So yes, we've got some different signaling events that are very similar in the 2, but we also have these populations. And then on the right, when you look at the normalized CT value, when we analyze the gene expression in these two different racial groups in the center of the tumor, and then also in the extracellular matrix center, we didn't really have any significant differences in the gene expression from the two groups. But when we looked at the edge of the tumor and the extracellular matrix at that edge next to the tumor, we did have significant difference. So something is different right there in that border. And for the study, we ran a parametric analysis. So we did some different statistical analyses. And one gene that kept coming up was S100A4. So we decided, okay, let's take this one step further and let's see what is happening with S100A4. And this is new data, and I'm just kind of showing a little blip of this, but what we did was we did with CRISPR-Cas9, we knocked down S100A4. We've actually done this in multiple cell lines, but I'm showing you here 231s and 468s, which are some of the more common triple negative cell lines that we have, where we have one that illustrates from European American and one from African American. And so let me take you through this. So what we did is, for this graph, is just showing the migration that we have of the cell line, okay? So we have our wild type, and then what we also did was we co-cultured with fibroblasts, right? And it's quite interesting how in that European American, just looking at the wild type with the co-culture, how this co-culture with fibroblasts, how the migration increased. When we compare this against the African American cell line, we didn't have that much of a difference between these two different parameters. Then when we look at the knockout cells in the 231s, it's interesting comparing the knockout versus over here on the 468, how we have completely different data. We've repeated this study 3 times, and we've come up, we didn't have any outliers. So this seems to be consistent, and we need to analyze this further. And our negative control, what happened with the negative control cells is these are just cell lines where we just expose them to the electroporation. So obviously this is having some effect on this activity. We need to go in and go through this a little bit deeper. But what we're finding is that we are having a difference in migration in these with the effect of S100A4. So again, my graduate student is taking these knockouts a little bit further, and she's also doing a lot of different cell culture assays, and she's added different cell lines as well. So we also have non-tumorigenic in addition to these tumorigenic cells. There's a review article with Bob Weinberg and his group, and this one was just put out in 2024, but it's a great read, where he is going through about dissemination and the process of EMT, and then the process of colonization. And my point in bringing this up is, we have this tumor, and the tumor is removed from this patient, and we have these disseminated cells that potentially could be tumorous at some point in time. We are not sure what is the signal that turns these on. And our immune system, in theory, takes care of most of these, which is great. Some of these disseminated cells, they can linger for weeks, for months, for years, for a lifetime. But then some of them also can start to receive a cue, and it leads to this colonization. And then we have what is known as metastasis. So again, this is showing, does this extracellular matrix, is this what's waking this up? And this is where I think a lot of our focus of our study needs to also go into this extracellular matrix. What are these signals that are turning this on? And I say here, what about phase 2 not shown? And that is that level of dormancy, right? So like, how long is dormancy actually happening? Can we reverse cells to go back into dormancy? That would be excellent. And a lot of this also is related to signaling that is happening in development. So if we can also figure out some of those early developmental cues, maybe we can also alter the signals that are driving these disseminated cells into metastatic potential. So another study that we've recently started going through is evaluating collagen and the collagen parameters in the extracellular matrix. Now, generally speaking, and again, this is generally speaking, African American women have higher density in their breast tissue when compared to other racial groups. Now, and I'm not saying that other racial groups can't have dense breast tissue, but it is more common in African Americans that they have this density. And Val Weaver is a person, she spent a lot of time where she's going through the different collagen parameters and what role that it has on tissue density. But we have learned that linearized collagen fibers are forming in a pattern where it actually allows cells, it basically paves the way, if you will, for migration. It increases cells' ability to migrate if we linearize this collagen in the extracellular matrix. So what we did was we had taken just some of our H&E samples, our slides, and we analyzed our slides using second harmonic generation, which is what you're seeing on these images. And we really weren't sure what we were even going to see. And these are just some early pictures that we had, and we thought, what is this showing? So when we looked at these samples on the top and then the samples on the bottom, and we played a game in the lab. So we would put a slide in there and we would ask somebody, what race do you think this is from? And it was interesting how we could almost figure out what race it was by simply viewing the arrangement of the collagen fibers. So we needed a way to actually analyze this. And so what we did, so what you do with this second harmonic generation is the way that we view this. And again, these are just H&E slides on a microscope. And you look at this forward-backward ratio, and you get this image, that black and white image that I've shown you. And then we utilized a program called Twombly, and we basically created a fiber mask where we could analyze the parameters of these different collagen fibers in these patients, right? And for each patient, we collected 20 random images. So the computer the microscope just kind of moved around the extracellular matrix and randomly chose. So we did not choose where the 20 images were collected. And we measured 10 different parameters, right? So we're looking at how do they branch, how closely are they packed, how do they linearize? And of the 10 parameters that we measured, which are listed here, area, lacunarity, length, and endpoints, 7 of the 10 were significantly different across these two racial groups, okay? Now, 6 of the 7 parameters were significantly higher in the African American patients. And 1 parameter was higher in the Caucasian or European American patients. And the one that was higher was lacunarity. And I'm going to go back a slide and show you, if you look here, so lacunarity is the second one. What lacunarity is evaluating is space. So the more space that is there that's not filled with something, the lacunarity value is high. So in other words, that's why this picture is super dark here for high, where these I would say are a little bit darker if they're low. So lacunarity meant there's more space. So the reason the African American was statistically lower is because they had more collagen in that extracellular matrix. So this was pretty interesting for us, and we're hoping to get this out and publish this soon. And now I'm moving on to our most recent data, and I've communicated this with a few of you. I've had this data for approximately 7 days. So this is very, very new. It's very fresh. This work was conducted in collaboration with Cold Spring Harbor Laboratory. So they have a core facility where they can conduct spatial sequencing. And what we did for this study is I had 4 triple negative tumor resections from African American patients. I'm sorry, 5 from African American, 5 from Caucasian or European American. These are all grade 3 tumors. And these are all tumors that are collected before any treatment. So there's no radiation or chemotherapy effect on these tumors. We subjected them to spatial sequencing. And unfortunately, and you see we've got a black line through 2 of these from each racial group. So with our initial spatial sequencing, 2 samples from each racial group failed our QC, which was really disheartening because we were pretty excited about our N equals 5. But I think we've overcome that. So, and with spatial, and here, let me show you this. So what happens if you look at this, I think this is a good image right here. Just if you're not familiar, and this is not single cell spatial, but this is spatial sequencing. So what happens is There's basically circles aligned all over the entire tissue. And the tissue is removed. You do a deep sequencing on that tissue. It's barcoded. So you have the data that basically assigns it back to that space. Now, again, it's not single cell, but it is telling you what the gene expression is in that one sample. And then we can backtrack and we can basically come up with a pretty good estimation of what the cell types are in that area, okay? So we did this, and one thing, and again, this is very fresh data, so bear with me here. But when we are looking at the collagen-rich clusters for the ECM and the epithelial clusters, and this is for our European American sample, we've got different high-end clusters here. Again, when we're working at the collagen ECM clusters, we've got a little bit variation for our African American samples and also for our EMT clusters, okay? So we're now thinking about also going in and even looking at receptor ligand interaction, trying to evaluate a little bit more on that tumor edge and where the tumor and this entire, microenvironment exist. So this is just showing traditional deconvolution results, and we had in here where we could establish where we have normal versus cancer epithelia. Specifically here is for EpCAM, which is great. But we really needed to address the issue of those two failed samples. And so what we did when we first got the data back, we immediately started thinking, okay, is there a different way to analyze this? And card deconvolution, this is an open source program that is available where it's a different way to analyze the spatial data. And what this does is it's basically looking at neighboring cells and trying to infer what cell types you might have that are close to that area, rather than looking at just clusters and what these different clusters mean. So we're trying to do this with our failed samples. And so the three images on the left are before we did this card deconvolution. The three images on the right are after. And basically it helps reduce a lot of the noise. It cleans up the samples. And we're able to now start to see, like here, we did not, and I'm gonna show another example, but this is afterwards where we're basically getting these hotspots, and we can do this for different cell types. So it's lighting up in different regions all around this tumor area. This is pretty exciting here. So here we have before we did this card deconvolution, and then once we refined it, we could show that where we've got this increased EpCAM expression in our tumor, and it's out there on that tumor edge, which is exciting. So now what we're going to do is go back through all the samples and do the card deconvolution on all of them to analyze and to see how this is going to vary our data. But again, a lot of ligand, I think some ligand receptor interactions will be quite helpful. This is another example of a low quality sample before, and then once we did the cleanup a little bit with this card deconvolution, we are actually having better data that is giving us that, again, we couldn't see it, and we're pretty excited that we don't have to just totally toss out those two samples. So this is one of these, and we put super strict thresholds on this. And these are all of these different areas that are now lighting up, where again, we did not see those before we actually did that card deconvolution, okay? So this is giving some highlights here where we've got obviously a lot of T cells, some luminal cells, some progenitors, some myoepithelial cells. So we're pretty excited about this data. So stay tuned for this, because again, this is relatively fresh. And this paper, it's with Tim O'Connor's group. They went through and they evaluated the ancestry lineage of a lot of these commercially available cell lines. And what is kind of interesting about this is if you look at, they went through just the NCI cell lines that were available. Look, the majority of these, so 59 of them out of 70 non-redundant were analyzed. Look at how many of these are of European ancestry, right? And compared to all of these different ancestry groups that we have. And so I have this bullet point on here about clinical trial history. Do you guys know what year women were added to clinical trials in the United States? Does anybody know? $1993. Yeah. Okay, so women were added to clinical trials in 1993. And when I say added to clinical trials, they agreed to join the clinical trials. They were part of clinical trials before 1993, but they did not consult to be a part of the clinical trial. And I have students, when they ask me that, they're like, well, then how did we ever determine, like birth control for women? And we actually used, there was a facility in Massachusetts where there were women that were part of this mental health institution. And so we utilized those women. We've utilized prisoners in the past. So just looking at just women in general, 1993 in the United States is when they were officially added to clinical trials, which is kind of surprising. And another challenge that we have in conducting a lot of this work with disparities is this one cell line and one gene, or when we're comparing two different cell lines. And I struggle when I submit grants or when I submit for a manuscript and the reviewers come back and they're like, you can't compare that cell line against this cell line because you're basically comparing just one patient to another patient. And I'm like, okay, but somebody can use one cell line and go into all these deep, beautiful assays for one gene, and they're published in Nature or Cell. And everybody's like, great paper, right? But it's just one cell line. So when you actually go in and you're like, I want to compare this cell line to this cell line just to see how they differ, people say, well, I don't know, you can't really do that. And another challenge is, in a lot of like that paper that we finally just got published at the end of January, it's a lot of the reviewers came back and you have N of 5 from each group. And I'm like, yes, but these are patient samples. I can't go get, just as many as I want. And my biorepository is great for helping provide tissues when it comes to their hands. But I have submitted for a grant and the reviews came back and they said, you can't compare these two. You need hundreds of cell lines from each racial group. Sure. Okay. That's doable. And that's what we're saying. Did a physicist or somebody review that? I'm not really sure who did that. And so determining ancestry is wonderful. And it's a wonderful way to go in doing a lot of the work that we do. But we also have to keep in mind and be mindful that we can't put people in one rigid category because we're all diluted versions of different racial groups. And so we're not all just 100% this or 100% that. So we also need to be aware of there's going to be people that maybe are a little bit, maybe someone has self-identified as one racial group, but genetically there's something else. Okay. And that's where, we need to move beyond this harsh categorization that we have. So in summary, so far for our work, we have established that there are morphological differences at the edge of the tumor and the edge of this microenvironment. And we have that gene expression difference that is going there. And then when we look at the density and the collagen patterns and the collagen arrangement, and it's interesting about this collagen study is this something that we could add when we do some sort of imaging and diagnostic? Can we view someone's collagen patterns and say, you might be more susceptible to cancer because of the arrangement of the collagen patterns that you have? And so this is something that's certainly worthy of further conversation. It's a nice non-invasive way of doing this. It's going to require some different imaging. But when someone goes in for their annual mammogram, can we also not just say, yeah, it's dense. Why is it dense? Is it because of the collagen? Is it the arrangement of the collagen? Are you now predisposed to potentially have more metastatic breast cancer? And then obviously we have this whole world of spatial sequencing that we've just embarked on, and we hope to certainly take this much deeper. I think this is phenomenal data that can really help us advance as we are analyzing the tumors and this entire niche that is surrounding these tumors. Like, what is this driving force? Evaluating tumors from different stages, evaluating tumors that are in theory quiescent or dormant, and potentially what are these cues that are driving outside of this tumor. And then we've recently changed our website for different reasons. But one thing that we have added to our website is we have added the word global. So I am working with Fulbright, and I am hoping to be a Fulbright US Scholar for the fall of 2026 for the entire semester. And the plan is that I will be actually traveling to Strasbourg in France. The way research is conducted in other countries, it's sometimes unique and sometimes it's quite beneficial. And the way that they conduct research, certainly biotech-related research in France, is they collaborate between research institutions. There is an institution of higher learning, like a university, and then there's also cancer treatment centers, translate large biorepository facilities. So my time in Strasbourg with Fulbright, the plan is what we are going to do is we're going to expand this ancestry and this diversity of the samples that we utilize. I'll be housed at the IGBMC, which is it's in Strasbourg. Also working with faculty, so there's a lot of clinical researchers at the IGBMC. There's faculty at the University of Strasbourg, and ICANS is a cancer treatment center, and they have one of the largest patient biorepositories, certainly in France, and it's one of the larger biorepositories that is housed in Europe. And so they have quite diverse samples that are there. They do also collect some self-identified ancestry information from patients when they take the samples. Believe it or not all biorepositories in the US collect this data. So you might contact them and say, I would like to get, these samples and these samples from diverse groups. And they're like, oh, wow, but we don't actually record that information. So, which is quite unique. So this is pretty exciting to take this initial work that we're doing, make it global, and see what kind of results that we get when we've actually expanded beyond biorepositories housed in the US. Finally, we can't do what we do without our funding. And these are just some pictures. This is my research guru, Bob Weinberg. I'm sure you guys all know them. But when I spent some time in Boston, I was able to interact with him quite a bit, and Brian Booth and Camila dos Santos, as well as some members of my lab. So thank you. Yes, you have the microscope or the micro? You have it? Oh, okay.
Is there, do you know anything that would, any lifestyle changes that could diversify the linearity of collagen? Or is that something that's genetic that there's nothing that can be done about?
Yeah, so, and I don't know, I'll repeat the question in case the people on Zoom couldn't hear. So the question was about lifestyle changes, and help me if I get this wrong. Lifestyle changes are something that we could do for the patterns of collagen and how they arrange. That is a phenomenal question. I don't have the answer. I definitely think that is something to investigate. Like, can we, so if we find out that someone does have certainly linearized, tightly packed collagen fibers, is there a way to cause them to not be so linear? I would think there would be, but it's a great question. Yeah. Yes. Okay.
Do them all. Great talk. Thank you for coming. It's nice to see you. So on that point, one of the things that we showed years ago was that NSAIDs could actually alter the alignment of the collagen in the mouse mammary gland. So that would be one interesting thing to look into. If you,
Is that a positive?
it would make it less tumorigenic. So it'd make it more curly,
Okay.
instead of that straight rigid collagen.
Yeah.
So that was more of a suggestion. So I have a question that I probably already know the answer to, but do you know whether the 5 patients that you were comparing ever had children or not?
Well, that's a great question. I do not know the answer to that. We are very limited on our patient data because I'm not conducting a clinical trial. I mean, these are just patients that are going through our cancer center. That's a great question. I can ask, so we did end up getting, we got the age and we got the smoker status. And it's funny, our biorepository, they're very hesitant to give us data because they're like, you could identify these people. And I'm like, I don't think so, but thank you. But we potentially could get that because that would be helpful, yes.
Yeah, I mean, you should be able to get whether or not they had a child or not, but, you know, we're interested in how recently they had a child, and I doubt that, like you were saying, some things just aren't collected, like crazily, that hasn't historically been, collected, but, when you go to do your studies in Europe, you'll have that data because of their medicine and the way they're tracked most women from birth to death, whereas here we move around and we don't have centralized, you know, records. So you might be able to get that data more readily in Europe.
Yeah, I'm pretty excited about that. Let's hope that Fulbright stays a
Yeah.
functioning thing between now and the fall of 26. Yes.
Fingers crossed. And
Yes.
Congrats on that.
Thank you.
A question from the Zoom from Paul Jenick asking whether ethnicity-based collagen differences are body-wide.
That's a great, I don't know. I don't know. That's a great question. We all need to get in the lab and start running some of these studies, right? Yeah, I do not have that answer.
Thank you. That was a extremely interesting talk.
Thank you.
Seeing the differences that you have in the ECM rearrangement, I know this is relatively new data, especially with your spatial sequencing. Have you given any thought to looking at that interface and seeing whether it's the tumor cell itself or the fibroblast or the macrophage that might be contributing to that rearrangement? I know either through direct interactions or through synthesis of different proteins, you can rearrange that. I don't know. Or something like MSI that might get at that. Have you considered probing that question?
Yeah, no, that's a great question. So what we're actually doing is we have handed some of our data and our slides over to a pathologist. He is now going through and scoring these slides as we did with the laser. So he's going to score these, and we're actually going to be able to have defined data on what is the tumor proper and what is outside of the tumor. Because I think that the fibroblasts and a lot of these, you know, these extra tumor signaling events in this niche are driving this as well. Yes.
Thank you.
Yeah. Sure.
Do you know, in the areas where the lacunarity
Mhm.
was lower, meaning there was more collagen versus higher where there was less,
Yes.
are you able to assess cellularity in those specific regions? Is there something that you can do to tie lacunarity to what is, if it's not collagen, what is there? Are there immune cells? Are there fibroblasts? Are there tumor cells or something like that?
Yeah, so that's where we actually want to go in, and now we want to go in and start analyzing what maybe we can find this out when we actually do the spatial on outside of these tumors, is to obviously pull out what's collagen and see what else is remaining there. Yeah. I know there's something there. I mean, we know it's not a void space.
Yeah.
So there's definitely something that is filling that space up. Yeah.
Thanks.
Okay, I have two database questions. So one, you saw that the ECM non-tumor was very similar to the tumor when you did the 4 laser microdissections. Isn't that odd, or is that what you would expect, given that, you know, it is further, at least theoretically, from the heart of the tumor than the edge of the tumor? And so I was wondering if you could expound on how I should interpret, I guess, the edge of the ECM being similar to the heart of the tumor.
Oh, so the outside of the ECM. Yes.
Yeah.
So the most further part. You know, I don't know, but one thing that we did is that we have much lower incidence of EMT happening in the center of the tumor. and happening over in as far away from from the tumor edge that we could be, which EMT is obviously driving proliferation, but then we've got potential metastatic potential.
Mhm.
So the center of the cell of this tumor could be, yes, there could be some drivers there, but we don't have a lot of signaling cues going on there. So I think a lot of that similarity, maybe we just don't have a lot driving. And again, that PCR panel that we did, it was all the genes that were selected that were highly implicated in EMT.
Mm.
We didn't go deep through that panel.
Okay.
So I think that's also why there's
Okay.
not the differences.
Okay, and a follow-up, or at least a second question is, when you showed the migration data with the co-culture with the fibroblasts,
Mhm.
comparing the African-American cell line to the European-American, it looked like migration was higher across the board in the African-American population. And so, if I remember that plot correctly, and so what, you know, do you have any insight on why the migration would just inherently be higher, at least from a baseline standpoint in individuals with African-American individuals?
Yeah, well, possibly there is, and again, so and this one is just migration, maybe this is part of why TNBC is more aggressive in African-American patients, because the cells have a higher increased ability to migrate, and then to disseminate, and then to colonize and then metastasize. So maybe that's part of it. Yeah. That would be my initial thought, but if somebody has something else on that one, yeah.
Yeah, I get it.
Yeah,
I
right?
got two now. Sorry, thank you. Oh, I'm like scared they're gonna, kind of going off that same question, I was also interested here that the fibroblasts seem to have no effect on the African-American cells. So I guess, do you have a comment on that? But also kind of the follow-up to that, I was wondering where the fibroblasts were isolated from, and maybe it's a difference of I bet this probably doesn't exist because I know the cell lines are so limited, but if there was a Caucasian derived fibroblast versus an African-American fibroblast, would that change what you're seeing here?
Yeah, and that's one thing that we've thought about, this is the source of the fibroblast, and what effect this is absolutely going to have on this. So that we definitely need to go a little bit deeper on this, because I definitely think that the source it very well could be a contributing factor. Yeah. And the other thing too is on a lot of these studies, so a lot of our studies were conducted just in 2D. And so our next step is that we are not only going 3D, but because of this collagen study, we're going to vary the density on our 3D. And I think that's going to be pretty helpful, where we have a low density and a high density 3D, rather than a lot of our 3D studies, it's just across the board. It's just one density. It's just set for everything. And I think that's also going to change a lot of the dynamics of these cells. So this is great, I would say, starting data for a lot of good next steps. But we definitely need to add in also to those density variations in 3D and see what effect it has on these, especially on this with this knockdown parameter.
In line with Lauren's question, when looking at these different cell lines, and I know, as you mentioned, they are limited to compare across, but do you see when you put them into mice, obviously there's a difference here, ex vivo or in vitro, when you're doing these analyses. Does the collagen arrangement, have you checked by H&Es if that recapitulates what you're seeing in your patient samples as you start to drive into the, cellular molecular mechanisms?
Yep. So all of this work that I have shown here is all in vitro work.
Yeah.
And we do have some collaborative in vivo work that is getting started. So we don't have those mouse models to show yet, but that is definitely next steps. Yes.
Thank you.
Yeah. Is that good? All right. Thank you.